Welcome, everyone. My name is Diana Wall, and I'm the science chair of the Global Soil Biodiversity Initiative. And today we're putting on a webinar, and we hope you all will be interested in this. It is a very important topic. What is happening in the Arctic and what is happening at elevation. And we have a group here of experts that are going to discuss this, but I'd just like to do a little bit of housekeeping first. First of all, this will be recorded. It is being recorded and you can find it on YouTube, and we'll be sending that out to you. And secondly, there are going to be questions. Please put them in the Q&A. If you have any questions at all, put them in the Q&A box, usually down at your bottom of your screen. And if we don't get to them, we apologize, but sometimes there's lively discussions and we just don't make it to every single one. So at this point, I'd like to introduce, I'd like to introduce our moderator. Amanda Colts is from Washington University at St. Louis. I really want to welcome her. We're very lucky to get her to moderate this. Amanda, take it away.
Thank you, Diana. Let me share my screen here. So thank you, Diana. Again, my name is Amanda Colts, and I'm an Arctic ecologist at Washington University in St. Louis in the US. And it's my pleasure to moderate our discussion today with a really fantastic lineup of soil scientists. Before I introduce our speakers, I'm going to provide some brief introductory context for the discussion. So as Diana mentioned, today we're discussing the biodiversity of Arctic and alpine soils. Now, although these ecosystems are characterized by long, cold, and harsh winters, they are still teeming with life, which is visible to anyone who's been lucky enough to witness spring or summer in these environments. So for contrast, this is what summer might look like. During the summertime, you can really see that these soils are rich in life and that they contain most of the same major groups within the soil that you would see elsewhere, and that they also have very complex food webs. Now, it's no surprise to anyone here that these ecosystems are also changing rapidly due to climate change. So, for example, this figures from the recent IPCC report, and it shows observed temperature trends over land between 1980 and 2015, with the darker red areas indicative of more extreme warming. Those lines at the top show the boundaries of the Arctic region. Now, it's clear here that the Arctic is warming rapidly and dramatically, and in fact, this area of the world is warming 2 to 3 times faster than the global average. Now, these organisms, the soil organisms, respond to that warming directly, but it's not only the warming that affects them, there are also indirect effects of climate change that they're experiencing. Some of these changes include thawing permafrost and extended growing seasons due to the warming. In some places, we're also seeing increased nutrient availability. There are changing precipitation and snow conditions. There's changing vegetation in terms of increased shrub expansion and also changes in tree line dynamics. And in some places, we're also seeing increased frequency and severity of wildfire. Now, how soil organisms and ecosystems in these Arctic and alpine regions respond to these changes is broadly relevant globally, because, for example, even though the Arctic only makes up 1/5 of the total terrestrial landmass, it stores nearly half of the global pool of terrestrial organic carbon. That means that even small changes in local carbon dynamics could have consequences at the global scale. Now, because of this, there's a lot of research from these cold ecosystems, and a lot of that research has been done in the context of understanding how both plant and soil responses to climate change will impact carbon and nutrient dynamics. Now, indeed, like elsewhere, soil biodiversity is sensitive to the direct and indirect effects of climate change and also to land use change that's occurring in some of these high latitude and high elevation areas. Now, in some cases, we might have a decent idea of how individual species or groups are responding to particular aspects of climate or land use change. But overall, a major challenge in an area of research is to develop a better understanding of how environmental change influences biotic interactions and what the ultimate consequences will be for ecosystem level processes. Now, our panelists today will be addressing some of these key themes, including the direct and indirect effects of climate change and land use change on the composition of soil communities, and on important biotic interactions that contribute to ecosystem functioning in alpine and Arctic ecosystems, including above ground, below ground interactions and plant soil feedbacks. So now I'd like to take the opportunity to introduce today's speakers. Today, starting things off, we have Frank Hagedorn from the Swiss Federal Institute for Forest, Snow, and Landscape Research in Switzerland. We also have Julia Seeber from EURAC Research at the Institute for Alpine Environment in Italy and the University of Innsbruck in Austria. We also have Paul Kardol from the Department of Forest Ecology and Management at the Swedish University of Agricultural Sciences in Sweden. And we have Stephanie Kivlin from the Department of Ecology and Evolutionary Biology at the University of Tennessee in the US. Now, our first couple speakers will start us off in alpine systems, and then we'll eventually move to the boreal and tundra ecosystems. Each of our speakers will have about 5 minutes to present, and then after they are all done, we'll have an opportunity to answer your questions. So please feel free to use the Q&A to put your questions, and we'll get to as many as we can during the discussion. So Frank, you can go ahead and turn on your video, and the floor is yours.
Okay, hello everybody, dear colleagues. I want to move you to the high elevation, to the cold, and I'll present you insight about the biodiversity and biogeochemistry in alpine soils in a changing climate. I move you to the cold, and these cold ecosystems experience a particularly strong warming, not only at high latitude, what Amanda told us, but also at high elevations. The Alps are warming approximately twice as much as the northern hemisphere. And this warming also applies for other mountain areas like Himalayas or other areas. This is largely due to changes in albedo, but this warming, of course, these are cold ecosystems, have a tremendous impact on ecosystems. These photo pairs here indicate or show evidence that the tree line is shifting upwards due to climate warming. The tree line shifts documented here have documented a rise of about 60-meter elevation during the last century. When vegetation is moving upwards, this also implies that all the microorganisms that are somehow associated with these plants are shifting upwards. So in this sense, the vegetation or the warming has direct effects on the organisms by affecting activity, but also indirect effects by changing the plant-soil interactions. Soil organisms also impact these vegetation changes because they provide the nutrients and the soils that are necessary for the plants to grow in these environments. And this is an aspect which is often forgotten by plant ecologists, but which is probably evident for or known for all of us. Our group assesses the direct and indirect effects of climate warming on below ground communities and biogeochemical cycles. And we primarily follow two approaches. We manipulate the climatic conditions in alpine environments, especially at alpine tree lines, and we assess the plant and the soil community changes along elevation and gradients, which can serve as a proxy for climate. I only have a few minutes of time. I just want to show you one soil warming experiment which we have conducted in the Alpine at the Seeber, translated is the silent mountain near the Rose. And at this site, we have conducted a soil warming experiment for 6 years with heating cables, and we have measured carbon nitrogen fluxes, but we've also measured fungal communities using 4x4 fiber sequencing. Just to show you Some results seen a significant shift after 4 years of soil warming, promoting species that are known to be nitrophilous, that are nitrogen loving, and others are getting suppressed. What we see in the ground, we can also see it in the protein bodies on the soil surface. For instance, Hydrophorus speciosus decreases with soil warming, whereas the nitrophilous species Lacarius rufus increases with soil warming. So we observe here a fundamental shift in the functionalities, which in turn will also impact carbon and nitrogen cycle. Maybe just as a take home, which goes beyond science and which is also important for stakeholders and what we may discuss about later on. Alpine soils are in a cold environment. They are usually very poorly weathered. Most of the carbon sits in the organic layer. There's very little stabilization by the interaction with minerals, and therefore they store large amounts of labile carbon. And this labile carbon is particularly vulnerable to the decomposition that can be lost as CO2 back to the atmosphere, but it is also prone to erosion upon disturbances. And this implies that this thin layer of soil can get rapidly lost. And soil preservation, in this sense, is of key importance to preserve this little thin layer which holds our life. And in this sense, I want to thank you and my colleagues living in the same environment that are also relying on soil biodiversity and the services of soils. Thanks for your attention.
Wonderful. Thank you so much, Frank. Julia, if you'd like to turn your video on and go ahead. Thank you.
Yeah, thank you, Amanda. So I'm a soil ecologist and I work mainly on macrofauna. So that is the invertebrates that are larger than, let's say, more or less 1 centimeter in length. My study region is the Eastern European Alps, and very beautiful region, as you can see here. And mostly it's the region around and above the tree line. So here we are dealing with another aspect of global change, that is land use change. For many farmers, it's simply not longer profitable to continue farming their alpine hay meadows and pastures. And once mowing and grazing has ceased, dwarf shrubs emerge. And these are woody plants, and as we all know, they produce litter of quite poor quality. So on these abandoned sites, we found significantly reduced earthworm biomass. And since earthworms, as we know, are very important for litter decomposition, this has then severe effects on ecosystem functioning. We saw in feeding experiments that earthworms are not selective feeders. They might have a slight preference for forb litter, but they also like to feed on the dwarf shrub litter. However, as we saw in life history experiments, their fitness is then highly affected. They grow slower, they are smaller, and they produce significantly less cocoons than when feeding on high quality litter. So in the end, abandoning the traditional farming practices that have been going on here in the European Alps for centuries has a direct effect on the earthworm population in these mountain soils. Also, the presence of livestock has beneficial effects on the distribution of earthworms in higher alpine soils. On sites where sheep dung, for example, is present, we still found a very high number of earthworms, even at the mountaintops. On the other hand, the food webs in these high alpine soils are very simple. So with a high amount of omnivory, we think it's for the soil and the invertebrates, they simply cannot afford to be picky up there. But coming back to climate change, we use elevation gradients as proxy to study climate change effects. Probably you can't see it too well from this picture, but because it looks kind of woody, but the transects are pasture transects ranging from 1,000 meters to 2,000 meters above sea level, at times even 3,000. And here, when looking at species composition, we see a very high species diversity around the tree line. So these are the sites around 2,000 meters above sea level. I mean, there are exceptions, as you see here, for the ants in this case, but most taxa do have hump-shaped distributions along this elevation gradient. This very high species diversity around the tree line we do not see only in the soil, but also on the soil surface. So we had pitfall traps installed at the same transects. And also here, species diversity drops after 2,000 meters above sea level. But since there is so little known about species living in and on the soils of higher mountain regions, we regularly find new species for the region, which is nice, as for example, this nice beetle here, which is a member of the family Cleridae, also this jumping spider. And I think that is something very general, that we just simply know too little about the soil's inhabitants in these high mountain regions. To study possible effects of climate warming, we can also use snowbeds, which we do. So snowbeds are depressions up in the mountain, as you can see on this picture, where the snow lasts for quite a long time. And the vegetation period is very short, especially, of course, here at the bottom of the snowbed. So the soil community that we had a look at here in those snowbeds is mostly composed of columella and borybatid which are members of the mesofauna, because macroinvertebrates, for them, it's simply the soils are too shallow, and especially for earthworms, also the vegetation period is too short. In this research for plants and microbes, we actually did find differences in community composition between top and bottom of the snowbed. These differences were even quite distinct. However, for mesofauna, we did not find any such difference, neither in abundance nor in community composition. And we think this is actually mainly due to many of these species being very generalist species, especially concerning their ecological requirements. So in any case, there are quite many effects of climate change, land use change on the soil community in these soils, and we will see in the future how that will be even more pronounced probably in the future. So thank you, and I give back to Amanda.
Thank you so much, Julia. Yeah, that was great. Paul, would you like to go ahead?
Sure. Thank you, Amanda. I will share my screen. Here we go. Yeah, we do most of our work in northern Sweden. South of the Arctic Circle, we're working in boreal forests, and further north, we're working in the subarctic tundra. Boreal forests are particularly important in a global change context because of the large amounts of carbon stored below ground. But boreal forests are also interesting because of the often continuous layer of mosses on the forest floor. The so-called bryosphere consists of green mosses all the way down to senesced and decomposing mosses. And the bryosphere mediates many of the linkages between above ground and below ground ecosystem components, including soil microclimate. The bryosphere hosts a complex moss food web, which is similar, but it's also a little different from the soil food web. This moss food web regulates nitrogen and carbon cycling. And here's an example illustrating how moss microfauna exerts top-down control on cyanobacteria that are associated with these mosses and which fix nitrogen from the atmosphere, and how moss mesofauna alleviates this top-down control. The moss food web is also important in litter decomposition, and that's because most of the litter actually decomposes in the biosphere rather than in the soil. Then we tested also these effects of moss food web structure under different scenarios of drought. Drought has strong direct negative effects on these ecosystem processes, but drought also affects the moss fauna, often negatively, but not always. And this then could influence the trophic interactions in the moss food web, which may then either amplify or counteract the direct effects of drought. Then we move up further north to the tundra. Here we focus mostly on the interactions between plants, soil biota, and the climatic context. For example, Clarissa Spitzer, she looked at the linkages between plant root traits, their associated fungal, fungi, and then how these root fungal relationships could explain plant-soil feedbacks. And just as the previous speakers, we also use elevation as a proxy for temperature. This works quite nicely, although soil temperature often correlates less well with elevation than air temperature. Then what are the challenges? Well, there's many, but one of them is to better understand how and when global warming affects ecosystem carbon cycling. So in our threshold project, we relate processes to organisms, linking plants and soil biota to the carbon cycling functions they drive. But organismal responses are also typically not linear for a variety of reasons. And this then could scale to thresholds and tipping points where a sudden change occur in the state or the response of an ecosystem. And therefore, we explicitly focus on non-linear responses in this project. To do so, we will link shifts in the composition of plant and soil community using across global forest alpine and forest ecotones, and link the changes in these community compositions to the carbon cycling processes. So that's in the field, but not everything can be studied in the field. So for some studies, we try to take the field into the lab. And here we're squeezing some new climate chambers into our building. And then we can expose like small little intact ecosystems to different temperature scenarios. But we can also test plant community responses to warming in the absence or presence of certain groups of soil biota by constructing our own little ecosystems. And that would allow them to test whether warming modifies or disrupts or decouples interactions among coexisting species of plants and soil organisms, and what then the consequences would be for the ecosystem processes they drive. So then I'd like to end with a couple of discussion points. First, yes, soil organisms do respond to climate change. We know that, but their responses are typically not linear. That makes it really hard to make longer term or larger scale predictions. We also know little about how tolerant soil organisms are to climate change, but some are probably more tolerant than others. And that could mean that currently coexisting species would not respond in concert. And then the decoupling of ecological interactions may further fuel the unpredictability. And finally, yes, we do need to make some more progress on linking soil organisms to their actual functions. And with that, I will stop sharing my screen and I will give back to Amanda.
Thank you so much, Paul. I just want to remind the attendees that you can ask questions of the panelists in the Q&A, and then we'll start the discussion after Stephanie's presentation, which is next. So again, yeah, feel free to put your questions in there, and then we'll start getting to them in a few minutes. So Stephanie, take it away. Thank you.
Thanks, Amanda. All right, so thanks, Amanda. Thanks for everyone attending and inviting me to talk about the work that we've been doing in my lab on how fungi respond to global climate change along elevation gradients. And as other people have introduced, mountain ecosystems are very relevant. They cover 20% of the land surface and are experiencing climate change at up to 3 times the rate of other ecosystems. And because of this, we can use them sort of as a canary in the coal mine to understand how microbes writ large will respond to climate change in the future. And so I do my work at the Rocky Mountain Biological Lab in the Colorado Rockies. And we try to assess how climate's affecting microbes through several different mechanisms. One is using elevational gradients as a space for time substitution, because lower elevations are reflecting conditions that higher elevations will see in the future. The other approach that we're starting to take, obviously with microbes, we don't have long-term data sets, but we could start doing long-term observations of a single site. And so we see climate change happening over decades. And then the third approach is using experiments like warming manipulations and snowmelt manipulations to directly understand how these drivers are affecting below ground communities. So first, I'll cover some of the work we've been doing along elevation gradients. And we focused on 3 different groups of plant-associated fungi, below ground, looking at arbuscular mycorrhizal fungi and root endophytes. And I'm cheating a bit to talk about above ground symbiosis with leaf endophytes. And all of these symbioses are nutritional symbioses for plants and can run the gamut from being parasitic to mutualistic depending on environmental context. And we wanted to assess if climate change is affecting these fungal groups directly via changes in temperature and precipitation, or indirectly via changes in plant host abundance, distributions, physiology or phenology, or even litter input quality. And this is important to make this distinction because as we're trying to project forward about how climate change is affecting microbes, we need to know if we can make those models based off of climate directly, or we have to think about interactions with other organisms when we want to project how microbes are responding to climate change. To assess these indirect and direct effects, we sampled almost 80 sites along 6 replicated environmental elevational gradients across 1,500 meters and up to 18 different grass species in the Colorado Rockies. And from this survey and multiple papers that we've done in this site, we see this consistent effect of plant hosts really driving the composition and abundance of these fungal symbionts. And here I'm just showing the variants explained of leaf endophyte composition, root endophyte composition, and arbuscular mycorrhizal fungi. And you can see these green circles often have larger numbers, indicating that plants are the main drivers of these fungal communities. And surprisingly, even though we have this large gradient, we're not seeing any effect of climate directly on these communities. So they never pop up in any of our analyses. So this suggests we really do need to understand these interactions between plants and fungi if we're going to model how microbes are responding to climate change correctly. Like I said, nobody has long-term or long enough term observations of microbes. And so I'm going to skip that, but I hope we can talk about it in the discussion. And I want to just touch on some of the work we've been doing at experimentally manipulating climate change drivers to really hone in on the direct effects of climate below ground. And I want to make this comparison because I think we can learn different things from studying climate change along environmental gradients versus experimental manipulations. So environmental gradients are great. You can get long-term responses. You can even think about eco-evo dynamics, but there's many confounding variables that co-occur along environmental gradients. So for elevation, you have climate changing along elevational gradients, but you also have differences in plant communities, you have differences in soil resources, and all of these things, because they're co-varying, sometimes they're hard to parse apart which of those is driving change below ground. Experimental manipulations, in contrast, are really nice because you're only manipulating one thing. So I can warm a plot. I know that that's the driver of any effects I see. But this is a short-term response, and we might mostly see ecological dynamics and not be able to capture evolutionary signals. So the comparisons that I'm drawing here is using elevational gradients as a proxy for climate change. We also have different sites that we know have different snowmelt dates historically, and we have different slopes and aspects that also melt out at different times. And we can compare that to experimental manipulations of snowmelt date and warming. And what we see is that when we look at the variance explained by all of these drivers, so how much each of these drivers is affecting fungal communities, we can see elevational gradients having a much larger effect on the composition and abundance of soil fungi compared to our experimental manipulations, almost twice as much of an effect. And this could be because there's long-term evolutionary selection on these communities, or it could be that there's a lot of confounding things that are happening along elevational gradients that are not just warming like we're trying to compare. But I think it's important to start the discussion about what we're learning from these different approaches. So in our warming experiment, it's almost as old as I am, so 29 years, I'm older than that, but 29 years of warming, and we're still not capturing as much variance explained below ground in the fungal community as we do with elevation for a proxy. Does that mean that we're getting it wrong if we're using these experimental approaches to project climate change effects below ground? Or does that mean that elevation is not a good proxy for warming? And these are discussions that I think we need to start having and pulling these different streams of data together as we go forward, predicting microbial response to climate change. And with that, I'd like to thank my collaborators and our funding sources, and I'll turn it back over to Amanda.
Thank you so much, Stephanie. Thanks to all of our speakers. Those were really wonderful, thought-provoking talks, and I'd welcome you to turn on all of your videos now, and we'll start the Q&A portion of the webinar. So to all the attendees, please place your questions in the Q&A, and we'll get to as many as we can during this discussion. Since there's a question directed at you, Stephanie, that I'm just going to ask right off the bat. It's a little bit less general than some of the other questions, but while people still have your figures in mind. There was a question about the Venn diagrams that you showed with the abundance associations, and whether it's right to conclude that one is driving, like one would be driving the change in others. So I was wondering if you could comment on that before people forget what it looked like.
Yeah, so those Venn diagrams are showing individual drivers like plants or climate or space and how much they affect the fungal community. And there is some overlap there, but they're not necessarily thinking about the interactions among those drivers. And so you should, I think, interpret those as how much each of those factors independently is affecting below ground communities.
Okay, great, thank you. And then I guess for several of you, there are a number of you working on both elevational gradients and on experimental warming. So I thought, to Stephanie's point about when we should be using different approaches to be asking particular questions about these soil communities is really important. And I'm curious if others of you have found similar effects whereby maybe experimental warming doesn't show such strong effects as elevational gradients. So I was wondering if each of you could comment on that. And also, Stephanie showed results with regards to the fungal community, but what that looks like for other components of the soil food web. See. Paul, do you want to start us off, since you had mentioned both experimental warming and elevation?
Sure, yeah, because it follows nicely what Stephanie showed in one of her last diagrams, sort of the the power of using different approaches for very different questions. Because yes, we also have done observational studies, but also we, at least for the gradient in the far north in the Arctic tundra that I showed, we found actually quite little or not so many sort of significant effects of soil fauna along that gradient. But I think that also relates to what Stephanie mentioned. It's not only temperature that changes there, because it's really hard to find or to come up with a very good sampling strategy along an elevational gradient, because there's always, at least in our system and probably in many systems, there's so much variation within elevations. So that's one thing. So for temperature, what we will be doing in our new project is specifically also looking at microclimate within elevations. So as I showed, sometimes the proxy elevation is a nice proxy for temperature, but not always, especially not for soil temperature. We see a lot of variation there within a single elevation. So I think that's something that could help to have more spatially explicit temperature measurements within elevations, rather than just using the elevation as a linear sort of temperature gradient. Maybe then one more thing, if you think about the different kind of questions to address, because I think for making longer term or larger scale predictions, I think these observational studies are very powerful still. But then coming from a different sort of scientific background, if you want to really understand how these things work, there's maybe even sometimes a more fundamental question than a directly applied sort of question that is directly relevant for policymakers. Then these small scale little manipulative experiments, which we've done a lot and which we like a lot, because you learn a lot sort of who is affecting who, and if the plant responds to the soils, is the soils responding to the plant, etc. So I think that's the way we want to keep going with these two different approaches.
Maybe I also add a short comment. I mean, we also study both direct effects via warming and elevational gradients, and usually the effects are much larger along the elevational gradient. I think the main reason is that we have to look at the soil. I mean, it takes decades or centuries to really change the soil properties, and I think our community diversity really depends on what type of organic layer we have, how much we have in the organic layer as compared to the mineral soil. And now we have these fancy tools to study diversity, etc. But we often forget, I mean, that they are primarily, in my eyes, driven what is in the soil. I mean, if it's a thick age layer or if it's directly a litter layer on top of the mineral soil, and this takes, I mean, decades to develop, and this is what you see along these elevational gradients. Whereas when you apply a direct warming effect, it's a matter of some years, and yeah, you may see the litter layer to change or the litter decomposition, but you don't capture that maybe the litter decomposes faster and forms a much thinner organic layer, which actually is the reason why the community is changing. Huh? But I mean, this is maybe the soil morphology point of view.
Julia, do you have any comments on?
Yeah, I would agree on that. I've never used experimental warming in the field, but we also, of course, use elevation as a proxy for temperature changes. And especially if you look at the gradient spanning like 2000 meters, there are a lot of changes. What we try to do is keep some parameters the same, like exposition and the type of, let's say, land use. That's why I mentioned that we use pastures along the entire gradient. But of course, we know that like soil organic matter increases and soil humidity increases while more or less soil temperature decreases with elevation. But I also agree with what Paul said, that soil temperature is not always a very good proxy, because here in Italy, we have those inner alpine dry pastures where the soil heats up really a lot. So this is not such a good proxy. But still, I mean, what else should we do? And we cannot like put those experimental warming experiments in all sites, and we have to use something.
Right. Yeah. And this, there's actually a question right now that sort of gets at some of those land use, the land use history about whether any of you have looked at the compaction of soils along those elevational gradients. And yeah, maybe Julia, you could comment to that, like what kind of, how the land use history and compaction from livestock or other pressures influence them.
Yeah, well, we know that soil compaction is, of course, important, but I mean, those are really mostly steep slopes. So the compaction due to some heavy machinery or even heavy livestock is not that much because they cannot be up there. So especially if I talk about soils above the tree line, let's say starting from 1800, 2000 meters and above, soil compaction is not really that much of a problem. It's just too steep there.
Mm-hmm. Yeah, Frank, did you have something to add?
Maybe I've shown the cow in my last slide. And in the Alps, I mean, you often have these cow steps. So we made a study about it. And I mean, actually, cows, they are always stepping at the same place. Huh? If they move along a slope, they do not step in between. And as soon as they have these holes, they always step at the same place. And these are actually hotspots in these alpine environments because they are dense, they are waterlogged temporarily, and they have a lot of nitrogen input, I mean, from the cows, but also from the slopes adjacent to it. And these are hotspots, and they have a very different community than the areas in between these hot steps. Mm-hmm. But yeah, I mean, this is maybe special for the Alps with these intensive Alpwirtschaft or Alp pasture things.
And so in addition to livestock, are there effects of natural compaction zones, like in relation to rock formation and things like that, and how those might affect soil biodiversity?
I would say less.
I would agree.
All right. I guess to getting one of the comments from just a couple minutes ago about, perhaps one of the reasons that we see such big effects along elevational gradients is that, that there are all these different elements of the soil that contribute. It's not only warming. And so, Paul, I was wondering, I don't know how early in your experiment it is right now, and perhaps if it's with the bryosphere and not soil with your environmental chambers. I'm just curious if you or other panelists have thoughts on to what extent we can manipulate some of those elements that we're missing from the experimental warming sites in the field. Like how much can we get at some of those missing elements in the lab?
Good question. The one thing that we always have difficulties with in the field, for example, is like moisture. Soil moisture, so that's the one thing that we have been working actually much more under lab conditions for these bryosphere studies that I mentioned, where it's very easy to manipulate or apply drought treatments, for example. And the good thing there was that it's very easy to sort of transfer like a little mini ecosystem intact into like a PVC core or something like that and make it into like a little mesocosm experiment. So, and then you can of course manipulate many other things as well. We've also done like nitrogen deposition studies on the bryosphere in the lab. So that's one thing. Another thing which I think is very difficult, and maybe Stephanie, you can follow up on that, is the difference in snow melt, which I think is very inherent to very many elevational gradients where sometimes snow beds are at lower elevation and then the snow melt is super late. And so even though it's warmer there, the growing season is actually much shorter. And that I don't really know how to get around unless you would actively remove it, but that's also logistically like a little challenging.
Yeah, I've never thought about manipulating snow in the lab. I'm also pretty adverse to the cold, so I wouldn't want to lab that cold. But I think your I think you're right, Paul. So we're seeing a much larger effect of snowmelt than warming on all of these processes. And I'm kind of curious if everyone's seeing that. And I think it's because snowmelt sort of triggers the phenology of plants and microbes. And so changing when that happens, either via warming or directly by putting a tarp on the soil and just increasing albedo and then snowmelts earlier, just sets the whole trajectory of that season earlier. And that's something that we are investigating with a different project. And I don't know to the extent to which we're sort of interested in the extent to which plants and microbes can handle differences in snowmelt date, and do they become decoupled? And so that largely is their phenology triggered by the same thing? So maybe plants are really sensitive to light, and whereas microbes would be sensitive to soil moisture, and we just don't know that yet. And so I'm curious to see where that whole field is going. And I think we'll, over the next 5 to 10 years, resolve that.
Can you comment a bit further on, so these effects of snowmelt, is that when you see really large effects, is that specifically with the fungal community, or do you all tend to see effects on other components of the food web as well, more with snowmelt than with warming, for example?
So I can comment about fungi that I've done directly, but all of the collaborators in the group at the Rocky Mountain Biological Lab is seeing a consistent effect of snowmelt date affecting everything. So insects, plants, below ground, macroinvertebrates, all of that is triggered more by snowmelt date than warming per se. And so that sort of puts in the question about how do we extrapolate the data we do get from the warming experiments? Is that correct if we're not accounting for this soil moisture properties like Paul was bringing up?
There's a kind of a related question that maybe Julia can start things off with about how soil mesofauna and macrofauna, how their physiologies might influence their distribution along elevation gradients, and maybe some of this plays into the snowmelt issue as well.
Yeah, well, we know that the physiology has to be adapted to those conditions up there. This is probably also one reason why macrofauna cannot exist in those really high alpine soils. We know their life cycles are prolonged. For example, for Calambula, they usually have like 2 to 3 generations per year, and then they have like a generation every other year. And also for Oribetid mites, they then suddenly have a generation of 5 years, because they simply have to adapt to those short vegetation periods and snow-free vegetation period. But we were quite surprised in our snow melt gradient experiment that we did not find any differences, not the species composition, but also not in abundances. So we also think that we read some literature that, for example, Calambula, they can use the intra-Nivean space to move also during the cold season. So I don't think we have to suppose that meso and macrofauna are like sleeping during the winter, let's say 10 months a year, but they are quite active also during the cold season. I mean, snow is somewhat an isolation layer, so the temperature is not that cold. And we also know from earthworms, even though we did not find them in the snowbeds, they can also live in like -5 degrees Celsius. So we always think that they don't like the cold, but actually they do.
I'm curious, since you mentioned the extended life cycles of these organisms in high latitude, high elevation areas, since that's a topic that we haven't touched on before. I know I work a lot with spiders, and that we have some evidence that they're being able to produce multiple clutches in a year where they weren't able to before. And so I'm curious if any of you have evidence for some of these life cycles changing or increased reproduction during the growing season for some of these soil organisms, given that when snowmelt is happening earlier, they have a much longer growing season. What does that mean? Not just in terms of composition and abundance, but also in terms of actual physiology of the organisms and fecundity.
Maybe I can add on that. I know it's specifically for millipedes, which have, I think they have quite some larval stages. and they just simply have to stop earlier and then take the next larval stage to the next year. And so then instead of 2 years, it takes them 3 years to become adult. So especially for millipedes, this is just not the problem. It's just an adaptation to the colder environment.
Does anyone else have thoughts on life cycles or changes in physiology?
I think it's a great question. For microbes, we are just starting to address that. Thinking about even microbial phenology is a new sort of forefront of the field. And so I will have those answers soon, but it's scary, right? What could happen is if the microbes are active longer through the growing season, they could be mineralizing more carbon and then actually creating a positive feedback to climate change. And so I think it's important that we figure that out soon.
Yeah. So one of the other questions, I guess, gets at some of this as well, whether maybe Stephanie, you could speak to this about whether subsoil warming will affect carbon cycling and organic matter persistence. And if so, if you can specify a few ways in which soil microbes in the subsoil at higher elevation will influence carbon cycling.
Yeah, I can say a little bit. So there's great work by Caitlin Hicks-Prees showing that deeper soils are responding to warming, not in alpine systems, but we've looked for that in these plots. So when we tore down the warming experiment after 29 years, we dug pits down to 1 meter deep and tried to understand if soil carbon was responding at those depths, and we didn't see anything. And so that's good in a lot of ways. We're not losing carbon that we're not tracking, but I don't know how site-specific any of those dynamics are because we haven't done enough studies across multiple ecosystems to understand the differences in top versus subsoil responses to warming. And so it's hard to, in my mind, make a global projection of what's happening at depth. And maybe other people have more thoughts on that too.
Maybe I have a more general comment. I mean, I think as compared to lowlands, high elevation and maybe also high latitude soils store much more carbon in the organic layer on top of the soil. So I think it's much, I mean, it's a smaller discrepancy as you find in lowland systems. So by heating or by warming, I think you capture, if you heat only the surface soil, you capture this. I mean, the deeper soils are hardly weathered. They contain not so much organic matter. So this is a difference to lower elevations.
Do any of you know if some of those, so that the deeper soils maybe in some places responding to warming, whether that old carbon is being respired directly by the microbial community or whether it's also making its way into the food web? I'm curious, I get a lot of questions about
I don't know, because I think it varies also so much where you're doing the research, because the places where we are doing our work in the subarctic tundra, there aren't really any deep soils at all. It's often just a very thin organic layer on top of rocks or boulders. So it's even difficult to take like a normal soil sample. As soon as you poke in, you hit a rock. So I think that's also the one thing what we like to do in the new project that I briefly mentioned is to indeed look at across different sites globally, like in a global network sort of structure to see how context dependent all these patterns along elevational gradients, particularly when you transition from woody vegetation into alpine vegetation are. Because I think you can learn a lot from that from just doing that by just observational studies across transects.
Yeah, sure. Yeah, to that end, I'm curious, since many of you do alpine research, to what extent these alpine systems can be compared to other kinds of alpine systems. So, for example, we have a question from Carol Frost that you know, a lot of these mountains have a lot of snow, and she's wondering whether comparisons from tropical alpine environments would be valid, since those environments often have less snow or no snow buildup, whether that would help disentangle effects of soil warming from the effects of snowmelt. Do any of you have experience in these tropical alpine environments, or have?
No, but I know from studies that the tropical mountainsides respond completely differently. And I think Paul's approach, and other people are also doing this, studying multiple mountain ranges at different latitudes, is going to move our knowledge forward, but that's just started, I feel like. So it's a really good point though, that you could basically use natural experiments and differences in snow melt or snowpack to get at these questions.
Yeah, I thought I've no experience in tropical alpine regions, but the one reason why we haven't really focused on that so far is, yes, you have less snow, or snow, you take away one confounding factor, which could be snow, but I think cloud forming and humidity, that's more of an issue, I think, in tropical elevational gradients than it would be like in higher latitude gradients. So you may swap one confounding factor for the other. But yes, I think that yeah, it would be nice to to compare them in that way, and then indeed measure a few of these potentially confounding factors and see how that could account for it.
So there are a couple questions that are relevant to you, Paul, since you work in boreal soils that store a lot more carbon. I was curious what your experience is with how changes in the soil community are influencing carbon cycling. And I was also wondering if you could speak a bit to your work with the moss and bryosphere, how those systems might differ from like a typical soil system that some of the other folks might work in.
Okay, I can start maybe then with these bryosphere ecosystems, because as I say, they do often form such a continuous layer of mosses on the forest floor. So a lot of these interactions, what happens above ground and how that affects things deeper down in the soil, is mediated by the bryosphere. What makes it different is that mostly, I think, particularly also the wet-dry cycles, because these mosses, they dry out very quickly, but they also are very tolerant to that. The interesting thing then, when you're studying moss food webs, what is also a little different maybe from most soil food webs, is that it's maybe more dynamic vertically, when some organisms live in water films on the mosses, others are living more in the dry space in the mosses. So when it's, of course, when it's drying out, they can maybe migrate down. But then the larger organisms, but the smaller organisms, they may just stay dormant, so to say, until the mosses will be re-wetted. So we haven't really done that so much, but I think it would be very interesting to see over time, dynamically during drying wetting cycles, how these food web compositions change. We have looked at carbon cycling in the bryosphere and the underlying humus layer, but we've not really been able to directly link that to the structure of the moss / soil food web structure. It would be another future step.
Yeah, those moss systems are so neat and so different. Even just taking a soil sample there, or a moss sample, it's just,
well, yeah. Because the one thing that we have often had difficulties with is where to draw the line. When you take a soil sample, like one of the pictures that are shown, it starts from nice green mosses, and it's really like a gradient from green mosses to brown mosses to dead mosses to humus to different kinds of humus. There's no sharp lines at all. So where does your soil start? It's quite arbitrary in that way. Or where does above ground that transitions to below ground? So that's really different in these systems.
Yes, definitely. Yeah, I'm curious. So Julia, there's a question for you about whether you see effects of winter tourism affecting the soil community, and perhaps there are multiple of you that can also speak to that. But Julia, do you have comments on I don't know, I'm not sure if there's increased winter tourism or just in general, how the history of tourism and land use have affected things.
Well, there's a lot of winter tourism, of course, in the Alps, and I have not myself specifically worked on it, but I do know of studies, and there is, if we talk about soil compaction, probably there is the most compaction because of those heavy machines that have to make the pists. Not the people themselves, but those machines probably have an effect. And it's also the longer snowpack duration that affects those soils, because they try to keep the snow as long as possible for the winter tourists. Even here now in lower elevations, where usually we would not have any snow left at the moment. So I cannot really answer specifically from my research, but I do know that there is a huge effect.
Yeah, thank you. And then there was another question for you, Julia, as well, but I'm sure multiple of you that are thinking about different groups within the food web maybe can comment on this, about what the criteria is for assigning specialist versus generalist species. And I know that's complicated, that we have very little information on the life history of a lot of these organisms. And so how do you go about defining the feeding relationships, how those feeding relationships might change along elevation and under different abiotic conditions? It's very complicated. And do you have thoughts on that?
So for the first part of the question, I think that's probably related to what I said about the snowbed community. And there we said that the generalist species would be one that is really widely distributed, that you can find in lowland soils as well as in high alpine soils. And we were quite surprised to find a lot of them. Some of those columbular species, they don't really care where they are. And then we would define a specialist species as one that is maybe alpine endemic, or that is specifically associated like with mosses or lichens. So it's probably, maybe it's from the ecological point of view, not a very, let's say, sharp conclusion on the specificity, but we try to understand if it's a really widely distributed species or not. And concerning the food web, I mean, we did that with stable isotopes, and we did some, let's say, mixture models, and it's the isotopic space, it gets smaller. Like, or let's say not smaller, but we have a lot of overlaps the higher we get. So as I said, they cannot be picky up there, so they just eat what they get. They also don't, due to those shallow soils, don't have that much space to move. And it's actually a lot of organic material. Maybe that's related also to that earlier question about the old carbon. We saw that soil material itself does not have any part of the food web. It's really simply only based on the organic matter that's in the soil up there. I could not define the age of the organic material, but I think it's very much based on that material for all the invertebrates. So food web chains are getting shorter the higher we get.
I just like to remind the audience to please put your questions in the Q&A if you have follow-ups on any part of this discussion or other topics that we didn't get to. We have about 20 minutes left. So if you have any last burning questions, please put them in. I just, with regards, so we touched a bit on abiotic controls and on plant soil feedbacks, but you know, some of this conversation is also getting at the extent to which higher trophic levels, and Paul kind of touched on it a little bit, like what kinds of species interactions might also be important here for controlling the composition and the activity and the abundances of these soil communities, and including the microbial community. And how might we start to be able to get at that, given that we still have this challenge of not knowing all that much about like the natural history. But if a lot of the organisms are generalists, maybe some of the interactions might be similar across these gradients, maybe. Now, do you have thoughts on other biotic interactions other than plant soil that might be important? Who might want to start us off? Paul, do you have thoughts on this?
I have to think about that. Other than, because you mentioned plant soil interactions, you're thinking about something above ground, you mean, or?
Oh, I'm thinking, I guess, like predator-prey interactions within the soil,
okay.
or you would, I guess, this, sort of like getting around it a bit. But you had mentioned, all these different organisms. A lot of them have nonlinear responses to warming or climate change, and how might some of those mismatches within the community? Do you think they might matter, or is it really more about the abiotic and bottom-up effects of the plant community?
Probably a combination of the two. The one thing that we have been wondering about but haven't really addressed it is competitive interactions within trophic levels of soil food webs, because we often like to depict soil food webs just as boxes, like one box per trophic level in a way, but there's lots going on within trophic levels, and there's a lot, which is always sort of a headache sort of thing in soil food webs, is the high level of omnivory, particularly when it comes to meso, macro fauna. Many of these organisms eat a lot of different things, as mentioned, just whatever there is, they feed on. So that makes it really complicated to disentangle, particularly if what we often do is take a soil sample and analyze a soil community, rather than just starting from individual species or individual organisms and then test what they are doing. Because that would, of course, be very neat studies if you would have like a whole set of cultures of all these different species and put them together in whatever kind of combination. But that's technically just so difficult to do. If you compare it, for example, to the studies with plants or so, where you grow different species together with or without a herbivore or something like that, which is technically just much easier to do, addressing the same sort of questions. But for soil food webs, that makes it difficult.
Right. So for your experiments that you're doing in your climate chambers, how is your group deciding which organisms to put in there? Which species interactions do you think might be important, whether those be competitive or predator-prey?
Good question. We haven't started them yet, so that's definitely something that we need to think about first. Now, of course, we probably start with something quite general, and then hopefully can narrow it down like along the way. Because another difficulty is, of course, if you want to use natural soil communities or soil food web compositions, how to disentangle like different groups, or how to separate them. Because there's, of course, studies that separate organisms based on size, like microfauna, mesofauna, macrofauna, by sitting, those sort of methods, which somehow distinguish between different functions, but size is not always correlated, of course, to their function or their trophic preferences. Predators are maybe typically a little larger, but not always. There's also very small predators in the soil food web, so they may end up in a different size fraction. So that's one real difficulty in separating individual or effects of individual group of organisms. So that will be a very good first step to think about further how to go about that.
I'm sort of curious in general. There's an idea, right, the stress gradient hypothesis, that under more stressful conditions, you get more beneficial interactions. So maybe mutualisms will become more important at the tops of mountains as opposed to the bottom, because it's a more climatically stressful environment. And I'm curious what people think about that, and if we can buy into that to say, okay, in the future, as these environments warm up, maybe you're going to see more competitive interactions than we currently do. Or is that too broad of a framework and we can't really project like that? I don't know the answer. I'm just curious what people think.
Frank or Julia, do you have thoughts on that? It's a great question.
It's a very good question. I would like to know the answer myself. I can't even tell. because there's so many changes going on then, because if you like do a little change, it has such a cascading effect in the southern ecosystem that I think we first need to understand many more of those effects to then really specifically answer that question. I am afraid, but maybe.
Yeah, maybe
I
can add on this. I think it depends where you are. I mean, what we have so far ignored is actually the development of new soil. I mean, in the Nivelle zone or from newly snow-free terrain where new soil is developing. I mean, this is a new area where organisms can also live and spread, whereas other areas lower at tree line, or they may respond very differently. So, I mean, we better have to differentiate where we actually study or where we look at warming effects and these kind of interactions. So it very much matters where you are.
Yeah, this is sort of related. A couple of you had in your slides mentions of pathogens, and my assumption there was that it was specifically with plant pathogens. But of course, there are parasites and other parasitoids and other groups that also have their life cycles in the soils. But I'm curious, I'm trying to remember, I think it was maybe Paul and Stephanie, I think that might have mentioned pathogens. If you could speak a bit to how plant pathogen interactions or other types of interactions with pathogens might be changing along your elevational gradients or with experimental warming, and what the big questions there might be. Paul, do you want to start things off since?
Yeah, I've also mainly just been thinking about root pathogens, because that's the thing that we have been mostly looking at, not directly in relation to these questions, but either fungal pathogens or plant parasitic nematodes. I cannot recall if there was any very strong relationship, for example, between elevation and certain plant parasitic nematodes, not that I recall. So for fungal pathogens, maybe Stephanie, you've done more fungal work, how they might change in response to warming. One thing, of course, if it's getting warmer, you might get different sort of balances between growth and defense in that way. So if you get higher nutrient availability, you get maybe faster growing plants, and you may invest less in defense against root or foliar pathogens for that matter. That could be one sort of starting point in framing your question.
Yeah, I think that's a great thing to bring up, that it's not just what the microbes are doing, it's how plants are responding to warming and how they change their allocation, either above and below ground, or like you said, defense versus growth. We've done a little bit in this warming experiment where we cultured fungi from leaves and noticed that their growth rates were faster under warming. And a lot of those tended to be pathogens, more so in the warming plots than the control. But that's far from conclusive about, I would hesitate to make a broad brush statement about that.
We have a question about, so in glacier foreland food webs, the importance of nutrients, detritus, and organisms blowing in from lower latitude or lower ecosystems can be important for determining processes and assembling those communities. And so Carol Frost is wondering whether subsidies from lower elevations are detectable in alpine communities, and whether anyone's looking at that. I know, and this also, I guess, gets to the question of to what extent we're seeing range expansion, somewhat related, whether we're seeing range expansion and whether we're even at a point where we can detect that in these systems. For Arctic systems, at least, it's very difficult because in a lot of cases, we don't even know what species are there. And there's not good monitoring of these areas. So I'm curious for these alpine systems that maybe not are more accessible in some cases, but in some cases they are, particularly where there's tourism and where there is some land use by people, whether we see evidence of organisms blowing in from lower latitudes or range expansion. Julia, do you want to comment on that since you've already spoken about land use a bit?
Yeah. Range expansion is really difficult, but because long term, as already Stephanie has already mentioned, also for invertebrates, we are really missing long-term data. We do have those LTER sites established, but I mean, they are not that old yet. And since soil ecosystems, they take such a long time to respond to any changes, it's really difficult to say after 15 to 20 years. And range expansion is something that I have not really seen yet. But I think it is there. So it's hard to comment on that. I agree with those glacier foreland studies. We do have one of these, and we saw those predator-dominated food webs right at the beginning after the glacier is melting, with those blown in detritus from all around as the basis. I mean, I don't know if we can compare this to any climate warming specifically. It's more a succession afterlife. Yeah, I think this is really difficult to answer in a very specific answer. Sorry.
Frank, did you have something to add to that?
Maybe I add to it, but not specific to soil communities, but I know it from the vegetation monitoring of mountain peaks. I mean, which actually indicate that you have an upward species shift. And they always face the problem that actually the tourists hiking up to the mountain peaks are bringing up the seeds in their shoes. And I mean, they contribute to this species distribution. And this will also be blown in seeds, but we humans also contribute to it. And of course, but the organisms, they also have to meet a certain substrate, I guess. Huh? I mean, which you don't have on bare land on the high elevation or the high mountain peaks. So it very much depends on what you get where you land. Huh? So I would interpret it in this way. Huh?
Yeah, I think those are great points. This is one of those questions that scares me, because how would we know for things that don't have long-term records for? How do we even know that their ranges are changing, or are they stable? I think there is some data, I'm forgetting offhand the citation, but I think there are some records of mushroom-forming fungi moving upwards in elevation. And so I think that's a signal that is telling us that we're missing a big part of the story when we're not thinking about, we just don't have long-term records to see that. A lot of other below ground.
Right. Getting it back again a little bit to these elevational gradients and what big effects we see along elevational gradients. There was a question that maybe Stephanie can speak to about whether you see similarly strong effects even going across gradients at the lower elevation. So if you're at 100 to 200 meters, do you still see changes in microbial communities, or do you really need those like really big gradients to see an effect?
You can see it everywhere. It's different effect sizes, right? And we've compared thinking about lapse rates in climate. And so if you have a warming manipulation of 2 degrees, what is the elevation range that you should be comparing instead of the entire gradient? And you can see differences even at 200 meter chunks all the way from shrublands at the bottom to the top of the mountain. I think it's interesting to think about how those effect sizes change as you're looking at different chunks of the mountainside, though. And that is something I have not done.
Well, one thing that we haven't touched on, I guess we only have a few more minutes, but so maybe just time for a couple more questions. But these systems are really difficult to access. A lot of the field work happens during the summer, but as a couple of you have mentioned already, that the wintertime dynamics might also be important. Of course, we're restricted by the academic field seasons and access, and in ways that perhaps folks that work in temperate ecosystems are a little bit less restricted. And so I'm curious whether certainly the pandemic has held us back in advancing a lot of field work, whether there are any kind of new and upcoming kind of technologies or methods that are helping us to better understand soil biodiversity in these remote and difficult ecosystems. Do any of you have thoughts on that? Have there been any good things to come out of this pandemic for pushing forward some of those needs that we've had for a long time already?
Are you specifically asking now for field work or for field technologies or anything?
Sure, for field technologies, for, I don't know, has there been like new network science that has like helped? Are people becoming more collaborative? What ways do you see for moving forward for developing more general understanding of these soil biodiversity big questions in these remote areas? Is there anything new and upcoming that might help us in a way that maybe wasn't available 5 years ago?
Maybe we have to add that the Alps here are not that remote. We have so many roads everywhere. So for us, it's not that difficult to have access to all those sites. So remoteness is probably not a problem for us. I can only add that here at EURAC, we do have something that is called the Terra X Cube. It's a new climate chamber that can also manipulate the pressure, and we can have a gradient up to 9,000 meters above sea level. So, but this is not something specific that came from the pandemic or something, but that has been worked on for quite some time, and now it should go online and work. For everything else, I mean, as I said, the Alps are not that remote, so we try to use the LTER sites as much as possible with very dense network of climate stations to have all the, let's say, the abiotic factors that we need to understand community composition.
Does anyone else have thoughts on kind of like new and emerging methods for looking at some of these questions that might have come up recently?
I mean, maybe I can add a funny one. I mean, is to make the use of microphones, I mean, to listen to faunal activity. So we have a guy working in our environment who actually uses super well-performing microphones and tries to link it to soil faunal activity. And in my eyes, this would be a way also to monitor activity in wintertime, or also in areas that are remote, and also continuously. Because I mean, I think this is what we lack. I mean, if we measure the abundance, we don't know what the guys are doing. And by having a continuous monitoring tool, this would be something cool to work on. But maybe you know about it already. So it's not a fancy DNA-based technology, but I think it's a thing, which can help to get a better temporal idea about what's going on in the soil.
Yeah, no, that's wonderful. I think that's what we need. We need some creative ways to start chipping away at this, particularly during times when we can't be there, when it's not feasible or inaccessible, either one. Well, we just have a few minutes left. And so Sebastian asked, I think, a very good closing question for today. So maybe each of you could just provide a comment about this, about what the major take-home messages are for Arctic and high elevation, the next generation of soil scientists. And so I will leave that to you, how you would like to interpret that, whether that be in priorities or what our institutions and governments need to do to help advance soil biodiversity research. But what would you say is the next, the big take-home message for the younger generation of scientists seeking to work in these ecosystems? Frank, would you like to go first?
Okay, maybe just to say, I mean, mountain soils are vulnerable because they have no stabilized carbon, so they lose carbon. I think we have to define where we work at because there's also an area where new soil is developing. So we are not only losing carbon by climate change, but we may also gain carbon because new soil is formed and new soil organic matter is accumulating. So we have to differentiate this. And maybe as a better take home, I mean, I think, our communities, maybe the soil community is working together, but we are working too little together with the plant ecologists that are kind of leaving the soil out, and we better have to link our two fields together. And this is a new area in my eyes, or better area, growing area, where new developments will come.
Thank you, Frank. Stephanie, do you have thoughts on last take-home messages?
I think my main thought is that we have done a lot of work, but there's still a lot of work to do to understand how soils are going to respond to climate change in any system. And mountains are a really good proxy for understanding that. And so I would encourage people to think about using that as a platform to get at these questions of global change, specifically with what we talked about, understanding how interactions are maybe becoming decoupled with climate change below ground, understanding evolutionary and ecological feedbacks between above and below ground ecosystems. A lot of these things are still really open and would make great careers for people if they're interested in those ideas. So go for it.
Julia, do you have last thoughts here?
Yeah, I think we still know too little about any species living in those higher mountain soils. We need to know more about them. And not only, like, as I am a macrofauna person, I should talk to mesofauna persons and microfauna persons. And we should really try to understand the entire community in those soils. And I think we need to make people aware of the specificity of the soils, that it takes such a long time to develop. And if I just take a handful of soil, I take away years and hundreds of years of development. And I think that is something that people simply often don't know.
Yeah, that's great and very important to consider. Paul, would you like to comment on last take home message?
Sure. A lot, of course, has already been said, and I think we should not forget that there have been so many excellent studies from all kinds of places, from all kinds of mountain ranges, which have already created a lot of new insight. But maybe for the next generation, it would be very nice if they could help us think how to generalize our understanding from each of these different or individual studies, because I think has become already clear in what we've discussed just here, how different the responses are based on where you are based, for example, based on the study system, the vegetation type, the range of your elevational gradient, etc., etc. So improving the that difficult word, generalization of these findings, that would be excellent.
Yeah, wonderful. Thank you. Yes, I think even in the previous webinars, context dependency is always a major theme, and it's always a challenge in ecology, and means that there are always lots of questions to be asking and answering. So with that, I'd like to thank our audience for the thought-provoking questions. I'm sorry if we didn't get to yours specifically, but thank you for attending, and I want to thank the panelists for your engaging presentations and this really great discussion. I'll remind everyone that the webinar has been recorded, so it's going to be available on the YouTube channel. The link to that channel is in the chat. And so with that, I'd like to thank you all again, wish you a great evening or rest of your day, and we hope to see you at the next Global Soil Biodiversity Initiative webinar. Thanks, everyone.
Thanks, Amanda,
Thank you.
for
Bye-bye.
moderating. Bye.
Bye.